Okay, let's start the class. After the class, you will continue with whatever you are doing. Okay. So in the last class, what we have started with the different types of classification system. And when we talk about the different types of classification, we have started with something called as a two-kingdom classification. Two-kingdom classification of further works has ultimately led into the formation of five-kingdom classification and a six-kingdom classification system, right? What we accept is obviously now from the point of studies, we are accepting the five-kingdom classification. Who has proposed this? R.H. Whitaker. According to him, what were the five kingdoms? Monera, Protista, Fungi, Plantae and Animalia. Talking about the most primitive kingdom, it is Monera. And as we talk about evolution, we know what is the sequence, right? Monera pra ki hoise Protista, and it is from Protista that all the three kingdoms has evolved, right? Next, what we have done is we have also discussed the characteristics of Monera, right? Some of the basic salient features of regarding the cell wall, regarding their nutrition, etc., etc. And what we have completed is we have talked about the 8 types of Monera. No, out of the 8 types of Monera, pasta poribo libo. So today we will be starting with the first one, that is eubacteria. Heading koriba eubacteria. I mean, we'll start with a diagram. Okay, I think this is something that we remember, no? Monera is a kingdom of Monera is a kingdom of prokaryotes, right? So what we are going to discuss is we just in the form of a diagram, we'll see the structure of bacteria, what are the contents of a bacterial cell, etc., etc. While we are discussing the bacterial cell, together with that, we'll be completing this part also, because later in a chapter called as Cell, the Unit of Life, you have to talk about the differences and the structures between a eukaryotic and a prokaryotic cell, right? So tatu tat ki koribo lagibo, cellot tumar eukaryotic cell structure or prokaryotic cell structure duita poribo lagibo, but the structure of a prokaryote is something that we are going to cover it in this class itself, right? So by the time we reach that chapter, it is only eukaryotic cell that we will be discussing. Is the point clear? Okay. Now talking about bacteria, how many types? As far as types of bacteria is concerned, no, shape and size and types, it's not like this that all the bacteria look the same. Belek types of bacteria ase, maybe you have heard something called as a rod shaped bacteria. Coccus type ase, Vibrio type ase, Spirulum ase, right? So what we are going to draw is a rod shaped bacteria. Why we are going to draw that rod shaped bacteria? Because that being the most common among the 4 types, right? ডাঙৰকে আঁকিবা Eko measure kori pelai akibo nalage. You don't have to count also. These are nothing but certain hair-like structures, bas. And some of the hair-like structures are shorter and some of them are longer, but that's all. ইমানে ডাঙৰকৈ আঁকিলোঁ মাৰ্কিং ক'ত কৰিম আৰু ক'ৰবাত No, circular dsDNA. dsDNA, double stranded DNA. পঢ়িম আমি সেইবিলাক পিছে। और एक पेरिफेरल निकले लेटरल मेसोसोम्स लिखा। That's all. Akilwa. Fimbriae, Fimbriae. Fimbriae. Over. Whole no? এটা গুটেখিনি পাৰ্টছৰ অকমান ইনফৰমেশনস জানিব লাগিব কেটামানৰ বেছিকে বাকিখিনি okay. So if you talk about a bacterial cell or if you talk about a prokaryotic cell, it looks something like this. What we can see is the different parts, first starting with something called a cell envelope, right? What is cell envelope? The coverings all together are called as cell envelope. In case of a bacterial cell, cell envelope or keta layers, say if you look into the diagram, there are 3 layers, no? The outermost layer is called as capsule, cell wall and cell membrane. The first point, it is not necessary that capsules of bacteria thakibo, right? Kisuman bacteria capsule thake, kisuman bacteria capsule na thake, but as far as the cell wall and the cell membrane is concerned, each and every bacteria will have the 2 layers, right? Now whenever a bacteria has a capsule, obviously it will give extra protection, right? Extra protection to whom? Obviously to the bacterial cell. Or junto bacterial cell of extra protection ase, to ki hobo? It will be very hard to kill those bacteria. Normally when we talk about bacteria bilake ki cause kore? Infectious disease kisuman cause kore, no? So if you want to cure that disease, what you have to do? You have to take certain types of medicines so that you can kill them, right? So killing them, tahatok ultimately mane if you want to kill them, then tahator ki hobo? Protection shield to gusabo laibo, no? So jar protection shield khon ase ba capsule khon ase, tahator control koribole okman problem, right? Aru jar protection shield khon nai, those are less infectious. Jar protection shield khon ase, shield will not be using the word protection shield, will be using the word capsule, right? Jar capsule ase ba capsulated bacteria are more infectious as compared to the non-capsulated, right? Okay, next inside portion, obviously we know the term called as cytoplasm, right? Tate iman ekhu nai. Next is coming to the genetic material. The genetic material, obviously it's not a nucleus. Prokaryotic cell or nucleus na thake, no? Nucleus boli ami kiho karone nako? Because you don't have the nuclear membrane. So instead of using the word nucleus, we have used a word called as nucleoid. Nucleoid is present. Nucleoid is also called as incipient nucleus. Eketai bostu ami pisot likhim ibla, right? Now when we talk about the genetic material in the form of ki, in the form of DNA or RNA, so you have to be specific. What is the type of DNA that the bacterial cell has? It has circular DS DNA. DS ki bostu will be studying later, something called as single stranded DNA and double stranded DNA, thik ase? Aru higher organisms bilakoto DNA thake, lower organisms bilakoto DNA thake, but the structure is different. If we talk about the eukaryotic cell, the structure of DNA is something like this, right? And this is helical, right? Eukaryotes of ki thake helical DNA thake, kintu prokaryotes of bacterial cell or ki thake circular. So C keteba circular ni likhi pelai okol CDS DNA boliu likhibo pare. So you have to know that word, letter C, right? Double stranded, single stranded, tumalke jeta genetics bhalke poriba, biomolecules poriba, you will become more aware of that stuff. Next, Coming to the words called as fili and fimbriae. If you look into the diagram, we can see 3 types of hair-like structures. What are 3 types? One is fimbriae, the other is fili, and the other is flagella, right? So, 3 type of hair-like structures. Talking about the word called as flagella, I think we know what is the function of a flagella. Eta bacterial cell ba jikono cell eta jodi flagella thake eta kam ki hobo? Locomotion, right? But that does not mean each and every hair-like structure will do the function of locomotion, right? Yate okol flagella locomotion kore. As far as fili and fimbriae is concerned, their main work is attachment. Eta bacteria has to get attached to a certain cell, certain object, then only they can cause the infection or they can stay there, right? Attach hoboleu to kiba organs lagibo. So we have 2 hair-like structures called as fili and fimbriae. Shorter kuntu, longer kuntu. Obviously, the longer is the fili and the shorter hair-like structure is called as the fimbriae. Next point about the hair-like structures, 3 types of hair-like structures, and what is the point of origin? If you talk about fili and fimbriae, they originate from the capsule, right? But if you talk about the flagella, tahat ko pura originate korise sason. We have marked it from the cell wall, but later we will study that one part of them always remains attached to the cell membrane also. Eta part, pisot ami korim. As of now in the diagram, we can see that it has arisen from the cell wall, right? Okay, next, apart from this genetic material, we have something called as plasmid, which is considered as an extra genetic material. A extra genetic material ketia lage? Remember a word called as sexual recombination. No? So sexual recombination actually hoy ki genetic material from one bacteria gets transferred to the other bacteria, and the most common type will be studying something called as conjugation. So conjugation process or eta bacteria pa antu bacteria le genetic material transfer hoy. Kuntu genetic material transfer, it is the extra genetic material that gets transferred from one bacteria to the other. Is the point clear? Details it will become more clearer. As of now, what is plasmid? Extra genetic material. Another point, not necessary each and every bacteria will have a plasmid, right? Kisuman thakibo pare, kisuman nathakibo pare. Next point is talking about the plasmid. There are different types of plasmid. Plasmid ki hoy? Genetic material. Tat kenukwa dhoroner DNA ase, kenukwa dhoroner RNA ase. Depending on that, we have types of plasmid. The most common type of plasmid that we talk about is something called as sex plasmid. Why we are talking about sex plasmid? Because we will be talking about reproduction in case of bacteria. Recombination, as I said, no, conjugation is a process through which the genetic material gets exchanged from one bacteria to another bacteria. Genetic material ki jabo? Plasmid jabo, right? So, eta bacteria plasmid na thake, eta bacteria plasmid thake. Plasmid thakar pora kole jabo plasmid na thaka. Later we will come across certain terms like male bacteria and female bacteria also, and we will be studying which are sex plasmid ase tak ami ki boli consider koru male ar jone receive koribo tak ki boli consider koru female. Ebilak pisot ami details ot korim, right? So genetic material, extra genetic material. Now in the form of granules. What is granules? Grain-like structures, no? So granules regarding they will store phosphate, they will store food, etc., etc. Talking about food, no? I think you know that there are certain types of bacteria that are photosynthetic also. Right? Not necessary that gutebilak bacteria heterotrophs hoy. Kisuman photosynthetic bacteria ase. Jedi photosynthetic ase, obviously carbohydrates to produce hobo. So hey carbohydrates to kobat store koribo lagibo, so they will be storing in certain types of granules. Next is a word called as vacuoles. In the chapter called as cell, we will be studying about vacuoles in detail. However, the presence of vacuoles is not a specific characteristic of all the cells, right? Kisuman cells or ase, kisuman cells or nai. To make a very, broad differentiation between a plant cell and animal cell, generally vacuole is a characteristic of a plant cell, right? Animal cells are both horu thakibo pare na thakibo pare. In case of bacteria, however, if the question is asked, is vacuole present? No, vacuoles are absent. Instead, what we have used the term is called as a pseudo vacuole, and pseudo is a word that we understand, no? False vacuole. First, it is also called as gas vacuoles, and as the name suggests, tat ki thakibo? Gas thakibo, right? Gas thakile ki hobo? Buoyancy help koribo. So it will be present only in case of aquatic forms. Bacteria bilak kot thake? It is ubiquitous. If you remember, we have discussed that term, no? So not necessary that they are found only in water. For those bacteria that are found only in water, they have to remain floating in the water. Tahata kune help kore gas vacuoles bilake. Baki bacteria gas vacuoles na thake. Okay. Next is a word called as mesosome. Right? So what we see is a structure, or in other word, we can use a word called as invagination. Invagination buzi maune? Invagination mane ki? Something that goes inside. That is what is called as invagination. Suppose you are talking about a membrane like this, right? Ekon dhoro ekhon membrane. A membrane kon যিনি অকণমান ভিতৰলৈ সোমাই আহে এইখিনি জেগা আমি কি বুলি কওঁ ইনভেজিনেশ্যন ইট কুড বি লাইক দিছ অলছ' এণ্ড ইট কুড বি লাইক দিছ অলছ' ৰাইট এইখন হ'ল এখন তোমাৰ কাৰ্ভড মেমব্ৰেইন এখন ৰাইট ছাৰ্ফেচ এৰিয়া অকল এইখিনি কিন্তু এইখন মেমব্ৰেইনৰ যদি এটা পাৰ্ট এনেকৈ সোমাই আহে তাৰ ছাৰ্ফেচ এৰিয়া বাঢ়ি গ'লে নাই right so this is what is called as invaginations of which membrane invagination of the cell membrane is also called as key plasma membrane right karon ami janu either it could be a plant cell or animal cell plasma membrane to thakiboi lagibo cell wall thakibo ba na thake kintu plasma membrane to hodai thakibo so here what we see is certain type of invagination called as mesosomes invagination from which layer from the cell membrane we have two types of mesosome and similarly we have the different function normally amak jeta questions ahe no directly question ahe what is the function of mesosome instead of asking the what is the function of septal mesosome and what is the function of lateral mesosome question in terms of mcq or entrance point of view mesosome or function so we'll be just remembering one thing okay before going into mesosome and what is its function mitochondria পঢ়িছা ন আগতে at least অকমান idea আছে how does the mitochondria look like single membrane and double membrane structure মনত আছে নে something like this right so we are talking about mitochondria is also called as the powerhouse of cell why is it called as the powerhouse of a cell কি আছে তাৰ ভিতৰত ATP কি হয় energy okay now which physiological process is related with the formation of energy আমাৰ cells বিলাকৰ mitochondria আছে mitochondriat ATP আছে give me one example of a physiological process that leads to the formation of ATP respiration right or in other way around we can also ask that why do we respire to get energy right respiration and actually hoy ki the food gets broken down right when the food gets broken down ultimately we will see that the ATP gets synthesized ATP to code store hoy ba code form hoy mitochondria bhitor what is mitochondria a double membrane cell organelle now ATP কেতিয়া ফর্ম কৰিব respiration হলে ATP ফর্ম কৰিব now the question is bacteria respire কৰে নে নকৰে living organism হয় তো bacteria respire কৰিব তাহাতৰ ATP ফর্ম কৰিব নকৰে obviously কৰিব তাহাতৰ mitochondria আছে নাই are you understanding the question right so what we are saying is whenever a cell undergoes respiration ATP gets formed in case of a eukaryotic cell we don't have a problem because ATP is formed in the mitochondria itself however in a prokaryotic cell a difference between eukaryotic cell and a prokaryotic cell membrane bound cell organelles are absent in a prokaryotic cell did you understand the statement membrane bound cell organelles are absent in a prokaryotic cell he karone to nucleus nai nucleus on membrane আছে নাই so membrane থাকা nucleus to code present hobo eukaryotic cellot prokaryotic cellot membrane নাই to nucleus নাই mitochondria থাকিব জানো না থাকে কারণ mitochondria কি হয় membrane bound cell organelles so ইয়াত to mitochondria না থাকে যদি prokaryotic cellot mitochondria না থাকে তো ATP formation code হয় বা respiration code হয় who is involved is the structure called as mesosome is the point understood so question ahe where does respiration occur in bacteria respiration occurs in the mesosomes in case of a prokaryotic cell or bacteria is the point clear so in general what is the function of a mesosome to conduct the process of respiration since they do not have mitochondria is the point clear but however in the diagram ami duta mesosomes dekhaisu no eta hol septal mesosomes aru eta hol lateral mesosome as the name suggests it's found in the lateral side or towards the periphery side ole ase he karone tak ami ki boli koisu lateral this word septal mesosome is related with the function of the mesosome we have been discussing the point that mesosome tar main function ki respiration but apart from respiration they are also involved in septa formation what is septa cell wall type right division type so eta bacteria jeta divide koribo binary fission hobo tetale ki hobo it will break down into two bacterial cell tahator majot ekhon division lagibo na lage so hey division ba septa khon kune form koribo this structure and so it is called as septal mesosome is the point understood next what would be the position of the septal mesosome eta bacterial cell jeta division undergo kore it's the binary fission which part divides first is it the cytoplasm or it's the genetic material Obviously, the genetic material. Remember this type of a diagram. That we have a bacteria, we have the genetic material. Next, the genetic material takes this shape, and later the cytoplasm also takes this shape, no? So ultimately, first thought ki divide koribo genetic material kini. So septa ekon form koribo lagibo na lage. So septa kune form koribo septal mesosome. So septal mesosome or location to kun kini near the genetic material. Is the point clear? However, from entrance point of view, septal mesosome or functions basic hudha nai. Question is, what is the function of mesosome, or where in bacteria respiration is conducted in the mesosomes. Clear? Okay. So I think we have almost discussed all the points. Okay. One point is left behind. Polyribosomes. We know the word ribosome. It is also called as protein factory, or it is that organelle where proteins are synthesized. In case of bacterial cell, ihotor protein synthesis hobo nohoy, obviously hobo, right? So quote hobo ribosomes. What is polyribosomes? Many ribosomes together attached to a particular type of RNA is called as a polyribosome. Later we will study the question at what is polyribosome. At that time you cannot be writing many ribosomes together is called as a polyribosome. No. At the RNA fragment thake, that ribosomes will lagi thake, and as a whole the structure is called as a polyribosome. We will discuss it later. But as of now, bacterial protein synthesis obviously because ribosomes are present. Genetic material ase what is the type of genetic material? Circular dsDNA. What is the function of mesosome? It is respiration, or ATP is formed in mesosomes. Now, okkoman points ami likhim. And what did I say about capsule? Not necessary. Each and every bacteria will be capsulated. Some bacteria could be capsulated, some could be non-capsulated. As far as infection causing property is concerned, capsulated bacteria are more infectious as compared to the non-capsulated. Junto diagram akila, tare gutei points we need to discuss. We have to talk in details about the cell wall, the plasma membrane, etc., etc. So first likha, ribosomes or polyribosomes likhi sara, tar bracket or eta bostu likha aso 70S. Last class of koisilu niki 70S or 80S, difference between a eukaryotic and a prokaryotic cell. Nai kora no? Or maybe I have spoken to the other batch. Huh? Bondho koribo le? Te fan bondho koro. Ekon lagibo? Na nalage? Nalage no? Ekon ok. Okay. Okay. Ribosomes are kotha koa nai niki? 16S, oh yes, in that, I mean, 6 kingdom classification or Rakoman question. Remember, we have talked about the sedimentation coefficient, right? Ribosomes, how will you study ribosome? You will extract it from the cell. How will you extract? Is it easy to extract? No. Why? Because it does not have a membrane. So how will you do it? You will go for a centrifuge, right? Centrifuge machine or sedimentation coefficient, right? So accordingly, heavier bila ki hobo, settle down koribo, lighter upor thakibo. Accordingly, we have two types, 70S and 80S. 80S, 70S, I will piece out details korim cell. So question I have, what is the type of ribosomes that you see in a prokaryotic cell? 70S. And what is the type of ribosomes in a eukaryotic cell? Mainly 80S, but in certain type of cell organelles you see 70S also. So both 80 and 70 in eukaryote, only 70 in case of prokaryote. The points likha. What happened? Okay. So, a structure called as plasmid. What is plasmid? The extra genetic material is called as plasmid. Extra genetic material we understand that it's not a part of the circular DNA. right? Juntu ami nucleoid likhisilu, nucleoid to genetic material thake. Plasmid and nucleoid ekei niki nohoy. It is called as extra. Next point, it can, okay, we'll come to this point later. It has the ability to replicate itself. Is this point clear? Eta cellot, eta plasmid thakibo. A plasmid etar pra dutao hobo pare, right? Duta ketia hobo when the process of conjugation will take place. It has to get transferred, right? Bacteria cell de gutekhini to di nidiye, so it has a ability to replicate itself. Next, it can be incorporated into the circular DNA as it can be transferred during conjugation. This line would be more clear when we will be discussing conjugation, but what we understand from this line is a part of the plasmid ki asle extra genetic material, protective bacterial cell or nijor genetic material to asei, right? Nijor genetic material khini ami ki boli koisilu, nucleoid. So a part of the plasmid can get attached with the nucleoid also, right? And if that is something that happens, then the new structure that is formed is called as episome. This line and this line means the same, right? Plasmid getting attached with nucleoid, the new structure is called as the episome. So question I have, what is episome? So if suppose we draw it like this, I think we understand circular dsDNA, plasmid, right? Next cell tooth, is the point clear? What is episome, right? A part of plasmid incorporated in the circular DNA or in the genetic material is this new structure is called as episome. First point, episome formation ketia hoy during the process of conjugation. What is conjugation? Exchange of genetic material or transfer of genetic material between two bacteria, right? Okay. Next, talking about the types of plasmid, it depends on genetic material, no? Genetic material ki thakibo, DNA thakibo. So beleg types of DNA, beleg types of gene thakibo pare. Whatever gene is present, accordingly we have different types of plasmid. The first or the most common that we will study, or the one that helps in the process of conjugation, is something called as F plasmid. Conjugation ki a part of reproduction. Since we are talking about reproduction, we have to talk about fertility also, right? Fertility by reproduction, everything is governed by genes itself, right? So plasmid, kuntu plasmid concerned with conjugation is F plasmid. F plasmid or ki thake? Fertility gene to thake. Is the point clear? Similarly, we have something called as R plasmid also. A2 plasmid or ki thakibo? Resistance genes to thakibo. Our resistance hole ki hobo? It will be hard to kill that bacteria or control that bacteria, right? Surviving capacity to tahator basic hobo. Similarly, col plasmid, Ti plasmid. Ekhini, however, iman nalage. All right, iman details of nalage. Kintu we understand that there is a method by which certain bacteria are involved for killing other bacteria also. Eta bacteria aru eta bacteria mariu pelabo pare, right? Kintu provided they synthesize certain type of substance that is called as colchicine, right? Ekhini iman nalage, kintu the first one lagibo. Ei baki tinta iman question aha nai. So this is all about plasmid. Next, So what is pili? Hair-like structure is called as pili. What is the main function of pili? We will be talking about during conjugation. However, the most important function is attachment, right? So duta bacteria, pisot ami diagram akim, just to have an idea, if suppose we are talking about this bacteria, we are talking about this another bacteria, tar nijor genetic material thakibo, tar nijor genetic material thakibo, plasmid thakibo, plasmid nathake exchange hobo lagibo, right? Genetic material exchange hobo lagile, they have to come closer to each other, they have to remain attached also. So they will have pili, they will also have pili. So what happens is, form kore. Etuk conjugation tube buli koy, and who forms this pathway is the pili. So what is the purpose of the pili? To do the or to go for attachment, right? Attach nohua ke ta genetic material yale ahibo nware etu, right? So attachment at kune help kore, pili help kore. Jihetu we are talking about conjugation, we are talking about reproduction. This pili is also called as sex pili, right? Ami likhim pisot. Next, have you heard something called as gram positive and gram negative bacteria? So we will be studying about two types of bacteria, gram positive and gram negative, depending on kisuman bacteria at a particular stain absorb kori, retain kore. Those are called as gram positive. Why we have to talk about this? We will be studying something called as staining in bacteria. Eta bacteria kami color korim. Bacteria to kami color kyo korim, because you have to study the contents of the bacteria. It's a very small organism. Tumi keneke porhiba stain no korile parts bilek to clear no hobo, right? So we have a stain that is called as a gram stain. Kisuman bacteria gram stain to loi pelai retain kori thoi diye ar tak ami ki boli kom gram positive. Kisuman bacteria, they will take in the gram stain but they will not retain it. Those are called as gram negative. So as of now, this much information is more than enough, right? So we have a particular type that is called as gram negative. Etu point about question ahe. Pili kartat thakibo only in case of gram negative. However, we will study later gram negative bacteria are more infectious. Karon swa pili ko pra arise hobo capsule or pra. Capsule thakile ki hoy infectious hoy. Is the point clear? Okay, hey khol likim ha. They have two types, longer and shorter, jeneke pili and so the longer pili are called Why they are called as sex pili? They are called as sex pili because they are associated with conjugation. Is the point here? So talking about those hair-like structures, duta naam disilu, eta hol pili aru eta hol fimbri, but now we see they are almost the same thing, right? The longer one is called as pili and the shorter one is called as fimbri. Pili aru fimbri kot thakibo only in case of infectious bacteria. So we have written a word called as pathogenic. I think we understand this meaning, right? Okay, we have another term, F bacteria. F positive bacteria or male bacteria. As I said, lower organisms bilakor male or female nathake, right? Higher organisms bilakor hai distinct male or distinct female thake, right? Eta organism, it might be a higher animal or a human being, male kun, female kun, we have certain morphological features that define. But if you are talking about the lower organism, bacteria is a single celled organism. Takno male female keneke describe korim, right? The factor that we will be concerned with is etu gene ase ne nai. Jedi fertility gene to ase, tethal tak ami ki boli kom male. F positive, we have talked about ki plasmid, no? So F positive, or we are talking about F plasmid, or we are talking about the fertility factor. Fertility factor to jedi ase, tethal tak ami ki boli consider korim male bacteria. Jar hei plasmid to nai, tak ami ki boli kom female bacteria. Clear? Yeah. Yeah. এই দুটা point to actually functional ভিতৰতে পৰে, right? But যেহেতু এইটো pointer ওপৰতে খালি question আহিছে, so we have written it like we have given more stress to this point. However, we know that the mesosomes conduct two functions, one being respiration, the other being septa formation. Septa formation during what? During the process of cell division. Clear?
What's that?
Structure. Yeah, write it down. Oh, I'll write a point. এইটো পইণ্টৰ ওপৰত কুৱেশ্যন অহা নাই কিন্তু জাষ্ট ফৰ দ্য চেক অফ ন'য়িং we have discussed a structure called a mesosome. Mesosome কি হয় invagination. কিহৰ invagination? এখনক কি বুলি ক'ম? Plasma membrane or a cell membrane, right? So this type of a invagination is also seen in case of a eukaryotic cell also, right? This structure or this invaded portion or this invaginated portion is called as cristae. Right? Mitochondria is a structure for him. Tete is the ভিতৰলে সোমাই গৈছে এই এৰিয়াটো কি বুলি কম cristae. So তাতো আছে invaginations, eukaryotic cell or invaginations আছে, prokaryotic cell or invaginations আছে. Clear? Main function of mesosomes কি লিখিছে to increase the surface area of the plasma membrane. Plasma membrane or surface area বাঢ়িলে কি হ'ব? ATP formation বেছিকৈ হ'ব. Right? হ'বনে নহয় কাৰণ ৰেসপিৰেশন কোড হয় ৰেসপিৰেশন মেসোসোমছত হয় সো যদি মেসোসোমস বেছি তেতিয়াহলে ৰেসপিৰেশন বেছি যদি ৰেসপিৰেশন বেছি এটিপি বেছি ক্লিয়াৰ Okay, nucleoid, genetic material containing structure without the membrane is called as nucleoid. It is a characteristic of a prokaryotic cell. What type of a nuclear material we are talking about? DNA, RNA, duita thakibo, kintu maximum ki thakibo DNA. What is the type of DNA? It is circular. In case of eukaryotic, it's helical, right? Next is double stranded or single stranded. Mane we are talking about the DNA. What is DNA? Nothing but a thread like structure. ঠিক আছে? So edal jodi thread hoy, eneke jodi single ke ase single stranded. Jodi edal thread hoy, eneke double ke ase, it is double stranded, right? So in case of a helical structure, no, we have something like this. So edal strand double hol ne nai. So dudal rosi ase ne edal rosi ase. Jodi edal ase, tetale single stranded. Jodi dudal ase, it's double strand. Later we will study the DNA and RNA could be of two types. Either it could be single stranded, either no, it could be single stranded, it could also be double stranded also, right? Okay, next is eto upor to question ahe histone protein or the basic one of the another major difference between a eukaryotic cell and a prokaryotic cell. Ami kisman difference janu no, tar bitorte histone protein is present in case of a eukaryotic, absent in case of a prokaryotic. As far as this protein is concerned, what is the function of this protein? Packaging of the DNA. DNA ki hoy thread like structure, right? In case of a human cell, DNA to almost 2 meter long hoy. এটা সৰু সেলত 2 meter long thread এডাল কেনেকৈ থাকিব cell to বিৰাট সৰু বস্তু and it is not only like this that it's only the genetic material that is present. Apart from the genetic material, different types of cell organelles are also present. So ইমান দীঘলীয়া DNA বা genetic material to কেনেকৈ ৰাখিব লাগিব pack কৰি পেলে. So hey packaging ত কোনে help কৰে? A specific protein that is called as a histone protein. It is a characteristic of eukaryotic cell. Histone proteins of function কি? এটা বহুত ডিটেইলছত পঢ়িবা তোমালকে packaging of DNA in eukaryotic cell, right? So histone protein present, histone protein absent. যদি present তেতিয়াহলে eukaryotic cell, যদি absent তেতিয়াহলে prokaryotic cell. Clear? Okay. Next likha cell envelope. Okay, so talking about the cell envelope, as we have already seen in the diagram, it consists of 3 layers. No? Specifically, the outer layer to distinct hoy, capsule. But sometimes the third layer of the caps or the capsule might not be present. Instead, a slimy layer could be present. What is the meaning of a slimy layer? Viscous substance eta, athar nisna bostu eta, right? Gel-like substance. So you will have the plasma membrane, you will have the cell wall, right? Common for all bacteria. Kisumanot the third layer present, tagami ki boli kom capsule, bacteria ki boli kom capsulated. Kisuman bacteria plasma membrane thakibo, cell wall thakibo. Kintu instead of the capsule, they will have a gel-like substance, and this is what is called as a slime layer, right? So etu kami capsulated kom, etu kami non-capsulated kom, right? So outermost layer or the cell envelope or outermost layer could be a capsule, could be a slime layer also. So we will be talking about this too. Eta yar points likhibo laibo. Capsule thakile infectious. Ne capsule na thakile infectious. Obviously capsule thakile infectious, right? Infection cause koribo pora capacity capsule or logot keneke related. Because that bacteria cannot be killed easily. But if you talk about a bacteria that has a slime layer, hey bacteria to pani re wash kori dile tar protection kin nai kiya hoi jabo. So jedi protection nai, genetic material to ami antibiotics di pele mari dibo paru. Is the point clear? So hey chart khonote likhiba ba jot likha aru capsule or kisuman informations or slime layer or kisuman information. বেশি নহয় এটা এটা So what we have seen, if you are talking about a bacteria, it could be capsulated or it could be non-capsulated. If it is capsulated, then capsulated bacteria has the disease causing property and they are called as virulent, right? Virulent or non-virulent, infectious or non-infectious, right? So virulent property ketia hobo, jetia capsule thakibo. Whereas if you are talking about a bacteria having a slime layer, it is generally non-virulent because that slime layer could be washed away easily, right? So could be removed easily. However, it protects the cell against desiccation and loss of nutrients. Loss of nutrients we understood. What is desiccation? Drying up, right? Hukai sabo pare no, lack of, normally bacteria bur kot thake, watery habitat, right? Pani thakile normally thakibo, pani naikia hobole ki hobo, they might die up, right? So desiccation bacteria. Out of all this, no, question has been asked about this virulent property and the word called as polysaccharides. Ebilakor sugar tugar bur iman mukhosto nokoriba, iman details of aha nei, right? ये वाला लाइन हो गया, ठीक है. Okay, what did I write? And provides ki likhesilo? Sticky and gummy character. Sticky and gummy character and virulent property. Huh? Absent থাকিব কারণ পিলি বা ফিমব্রে arises only from the capsule. When we are talking about the cell envelope, if you go by the sequence, the outermost layer is the capsule, the next is the cell wall, and inner to the cell wall is the plasma membrane. However, when we are writing it down, plasma membrane information kom, hekhane ami agote etu likhilo. Cell wall okman details ot poribo lagibo. This part is over. Second dot likha mane does not mean capsular tolot plasma membrane ase. Capsular tolot hodai cell wall ase. থাকিব না থাকিব লাগিব হা it will not lage, you will get confused. Okay, what is the plasma membrane? Plasma membrane is also called as cell membrane. It is something that is present in all types of cell. It could be a eukaryotic cell, it could be a prokaryotic cell also. How does the plasma membrane look like, or what is the structure? Here we are just drawing a rough diagram. Right? এতিয়া এইটো ইমান ভালকে পঢ়িবও নালাগে কাৰণ সেলত পুৰা ডিটেইলছত পঢ়িম কাৰণ সেল when we be talking about eukaryotic cell তাতোটো plasma membrane থাকিব and how does the structure look like. Various scientists from time to time has given different models, but as of now আমি কি জানো whenever we talk of the plasma membrane it has a phospholipid bilayer তাৰ মানে it is made up of কি, it is made up of phospholipid molecules or proteins, right? So phospholipid bilayer কিয় কৈছো এইটো এটা layer দুটা layer আছে ন so এই কাৰণে phospholipid bilayer আৰু কি থাকে different types of proteins থাকে so if you look into the diagram একদম rough diagram এটা আঁকিছো almost 3 types of proteins দেখিছো এনেকুৱা type আৰু এনেকুৱা type right এই proteins বিলাক কি হয় right we have different names for that কিছুমান proteins if you look into the diagram এবিলাক কোন বাহিৰলৈ আছে ন these are called as external proteins or extrinsic পিছত আমি পঢ়িম এবিলাক হল intrinsic protein আৰু এবিলাক হল transmembrane protein যাৰ থ্ৰুৰে materials from one cell transports to the other cell right details এটা একো নালাগে as of now plasma membrane is something that is present in both eukaryotic and prokaryotic cell. It is made up of phospholipid bilayer and proteins. Clear? Phospholipid মানে কি এটা molecule যত phosphorusও আছে লিপিডও আছে দুইটা একেলগে থাকিব right so আকৌ পিছত আমি পঢ়িম এইটো এটা phospholipidৰ molecule right এইটো head part কয় এইটো tail part কয় পিছত ডিটেইলছত পঢ়িম right তথাপি লাগে যদি So phospholipid will have two parts, one a head part and the other tail part. Detailed description in another chapter. Just to know, we have a head, we have a tail. Piece of question ahe. Which tail is hydrophilic and which tail is hydrophobic? Hydro, we understand we are talking about water. Philic or phobic? You like someone philic. You don't like someone that's phobic. Means this part likes water, so it will take in water. This part does not like water, so it will not give out water. Right? পিছত question আহে এইটো চ্যাপ্টাৰৰ পৰা কোনটো পাৰ্ট হাইড্ৰোফিলিক কোনটো পাৰ্ট হাইড্ৰোফোবিক পিছত পঢ়িম আমি এইবিলাক ওকে লিখা আকা একো নাই এনেকুৱা জাষ্ট টেডপোল টাইপ দি দিয়া ইটছ নট আ টেডপোল হাউএভাৰ ৰাইট ভালকৈ আঁকিব লাগে ইমান বেয়াকেও ডায়াগ্ৰাম না আঁকে আৰু মানুহে দেই কোনোবাই হা সেইটোৱে কথাটো হ'ল এইটো টেইল নহয় হা টেইল বুলি কৈছোঁ টেইল ডাজ নট মিন ডেট তাৰ মুভমেণ্ট হ'ব ৰাইট পাৰ্টটো যিহেতু সেই ডাঙৰ পাৰ্টটোক হেড বুলি কৈছে ছেকেণ্ড পাৰ্টটোক টেইল বুলি কৈছে হা তাৰমানে লিপিড পাৰ্টটো লাগি আছে ন সেইকাৰণে তাক টেইল বুলি কৈছে মুভমেণ্টৰ কাৰণে নহয় ট্ৰান্সপৰ্ট অফ ছাবষ্টেঞ্চেছ ফ্ৰম ওৱান চেল টু দ্য আদাৰ চেল ইফ ছাপ'জ উই আৰ টকিং এবাউট দিছ এজ আ প্লান্ট চেল ন এইটোৰ লগত অকল এটা চেলটো নাই ন ইয়াতেতো আৰু এটা চেল আছে নাই জানো সব সেলস একেলগে লগ লাগি নাথাকে জানো তো এ সেল এ পৰা সেল বি লে বস্তু সাবস্টেন্সেস বা ইয়াৰ পৰা ইয়ালে আহিব লাগিব না লাগে সো ট্ৰান্সপোর্ট কিহৰ থ্ৰুৰে হ'ব প্লাজমা মেমব্ৰেন সেল ওয়ালৰ থ্ৰুৰে হ'ব আলটিমেটলি ভিতৰত কিহেৰে সোমাব প্লাজমা মেমব্ৰেন খন দিয়েটো সোমাব ৰাইট সো এট দ্যাট পইণ্ট উইল বি টকিং এবাউট অল দিস হৈ গল সো প্লাজমা মেমব্ৰেন ইজ মেড আপ অফ phospholipid bilayer aru proteins same type of proteins na different types of proteins different how many types of proteins 3 types of proteins very good next likha cell wall plasma membrane aru eta point likhi thoi dia mesosomes are present same sub same lies between the capsule and cell membrane হেবিলাক আৰু মুখস্থ কৰিব নালাগে we know the diagram so we'll be able to write it down right so it's semi rigid structure main function it prevents the cell from expanding and bursting expand কেতিয়া কৰিব যেতিয়া হি absorb water কৰিব right অনবৰত যদি absorb কৰি থাকে তো it might lead to the bursting of the cell in other way around what is the most important function of a cell wall to maintain the shape and size yes or no cell wall থাকিলে এটা particular shape থাকিব ন যদি cell wall খন নাথাকে তাৰ তলত কি আছে প্লাজমা মেমব্ৰেন খনটো বেলেগ বেলেগ শেপ লৈ লব পাৰিব right okay next from bacteria point of view if you are talking about the cell wall we can see two different types of cell wall one being of the gram positive bacteria and the other being of the gram negative as I said bacteria could be of two types gram positive and gram negative depending on what depending on whether they can retain the gram stain or not right কিছুমান bacteria color কৰি দিয়াৰ পিছত ধুই দিয়াৰ পিছতো কালাৰটো যে ৰৈ যায় কিছুমান বেক্টেৰিয়াক যে ধুই দিয়াৰ পিছত কালাৰটো ৰৈ না যায় কিবা এটাটো ডিফাৰেন্স থাকিব লাগিব ন সো দ্য ডিফাৰেন্স লাইজ ইন দ্য স্ট্ৰাকচাৰ অফ দ্য সেল ওয়াল ৰাইট কিহেৰে বনোৱা আছে সো উইল বি টকিং অ্যাবাউট দিস ওয়ান এন্ড দিস ওয়ান ইন দ্য ফৰ্ম অফ আ চাৰ্ট এখিনি যদি হল ইয়াৰ ডায়াগ্ৰামটো আৰু ইয়াৰ ডায়াগ্ৰামটো আমি আঁকিম এতিয়া এখিনি হৈছে নেকি দিস পাৰ্ট সো গ্ৰাম পজিটিভ আৰু গ্ৰাম নেগেটিভ সেল ওয়ালৰ ডায়াগ্ৰাম again this part diagram part to okol understanding or karone iman details ot aha nai kintu duta man point important moi kodim Okay. Now, as I said, the diagram is not important. What is the word that is very important is this word, right? Jetia ami cell wall ko, if you talk about cell wall, we understand that we are talking about a plant cell, first point. Second, cell wall is made of, in case of a plant cell, as I think I have discussed before also, cell wall is made of cellulose, right? But we will see that fungi also has a cell wall, but it is not made of cellulose. Right? Thik teneke bacteria also has a cell wall, but it is not made of cellulose. So instead of cellulose, what they have is called as peptidoglycan. So jeneke plant cell buli kole ba plant cell wall buli kole, cellulose is one thing that you will remember. Bacterial cell wall buli kole, peptidoglycan is all that you have to remember. Peptidoglycan is made up of the peptide part and the glycan part. This is nothing but accumulation of certain sugar, and that is the reason why we have written a word that is called as polysaccharide before. Right? So cell wall kon kihere bonua ase, different types of sugars ere bonua ase, right? Instead of using those words, this is a word that we remember, right? Now talking about the two different types of bacteria, as I said, kisuman gram positive, kisuman gram negative, kisumane color to retain kore, kisumane color to retain no kore, depends on what? Depends on the structure of the cell wall. If you compare what we see, the amount of peptidoglycan is more in this case, thinner in case of this. The next point of difference is, in this case, cell membrane or layer eta, but in this case they will have an additional layer above the peptidoglycan. So bilayer hoy at a single layer hoy. Ebilakor karone ultimately they fail to retain the the crystal violet. This is nothing but the stain that we are talking about. Right? Once it retains the stain, but bacteria to color ki hobo, violet ba purple, aru ihote jodi retain nokore, so eta bacteria jodi ami color disu no, color to kiho karone disu, so that we can study the contents properly. If suppose it retains the color, it will become easier for us to study. Kintu what about the gram negative? Tahante to retain nokore. Jedi ni retain nokore color to, does that mean we are not going to study the gram negative bacteria? No, we are going to use a different stain. Right? So we will be using safranin, and ultimately the cell will turn pink, right? Okay. So as I said, what is the most important word? First, peptidoglycan. Second, additional layer or covering. It has an extra covering. Third is this word, a particular type of acid that is present only in the cell wall of gram positive bacteria. Later what we will do is we will be comparing gram positive and gram negative. Presence of teichoic acid. Teichoic acid is also called as lipoteichoic acid. Presence of teichoic acid is a chief characteristic of gram positive and gram negative. It is the chief characteristic of gram positive, right? Thik teneke jeti ami gram negative porim, gram negative oru kisun important points thakibo aru gram positive oru kile duitare common point ki, in both the cases the cell wall is made up of peptidoglycan. Clear? Eta teichoic acid or kam ki ekhini hol niki? Acid to eneke likhe janu ne ami? So one of the chief characteristic of a gram-positive cell wall, the presence of teichoic acid. As far as the function of teichoic acid is concerned, functions are generally questions aha nai. That is the reason why I have not marked it down, right? So teichoic acid or question ki hai, it is a characteristic of gram-positive and gram-negative, gram-positive. However, teichoic acid or function ki, we understand. First, it acts as a receptor site. Receptor mane? Someone that receives, no? So, we'll be studying about viruses also. Virus is also a microorganism, bacteria is also a microorganism. Virus or nijor property ase. Virus or one of the property is it can become associated with any type of a substance, any type of a living being, etc. etc. Virus ko GI thakibo. Virus to first GI thakibo jeti eta host pabo, right? Second, living thing, virus living and non-living, it could neither be considered as a living nor it could be considered as a non-living substance also because of certain characteristics, right? Later we will study is a kisuman virus e ki kore bacteria attack kore. So eta bacteria tat jodi virus to lagi jabo lage kuntu part ot lagibo, it is in that area where the teichoic acid is present. Is the point understood? So we'll be, have you heard a word called as bacteriophage? Etu diagram dekhisa niki? এনেকাটা দেখিছিলা এটো কি হয় ব্যাকটেরিওফেজ ব্যাকটেরিওফেজ তার মানে ব্যাকটেরিয়া প্লাস ভাইরাস টুগেদার ব্যাকটেরিওফেজ রাইট সো ইয়াতে কি হ'ব যে এটো এটা ফেজ পার্টিকল কাক অ্যাটাচ করব ভাইরাস হয় অ্যাকচুয়ালি ফেজ বুলি কলে ভাইরাস হয় দ্যাট উইল বি অ্যাটাকিং আ ব্যাকটেরিয়া রাইট আ ভাইরাস দ্যাট অ্যাটাক্স আ ব্যাকটেরিয়া ইজ কল্ড এজ ব্যাকটেরিওফেজ সো এটো কি হয় ভাইরাস কেনেকুৱা ধৰণৰ ভাইরাস ব্যাকটেরিয়া অ্যাটাকিং ভাইরাস রাইট. So, coat attachable in this site, and second, it also prevents the degradation of a cell. Jate niki bhangi na jay. However, that's not important. So, ekhini to hold. So, main point ki peptidoglycan. First, teichoic acid. Second, third, extra layer. Extra layer coat ase gram negative what. Peptidoglycan or one point eta likha. What I said, peptidoglycan is something that is present in bacteria. But etia ki likhise aso? Almost all. Bacteria ki hoy eta monera, right? So we'll be talking about another bacteria called as archaebacteria, right? Beleg types of bacteria porim. Kisuman bacteria peptidoglycan na thakibo pare, right? But the normal or the common or the true bacteria or eubacteria tahator tat ki thakibo hoday peptidoglycan present hobo. কি হল সব হৈছে ন next word that is important is the word called as murin. As I said, kisuman bacteria peptidoglycan present, kisuman absent. So question ahe murin present in or murin absent in. So murin mane ki peptidoglycan. Peptidoglycan er duta part, peptide part or glycan part. 5 minutes extra আজি বিৰাট খং উঠিছে চাগে ন সবৰে খং উঠে উপায় নাইকিয়া কি কৰিম I am used to that students love me a lot because of this একটো লিখাই বহুত বেছি ন তাৰ ওপৰত এক্সট্ৰা ব্ৰেক টাইমটোও খাই দিয়ে I know all the curses Okay, বহুত লিখিলো mark কি কৰিলো murin right so peptidoglycan is made up of কি amino acids if you look into this ইয়াতো কি আছে amino acids আছে ইয়াতো কি আছে amino acids so peptidoglycan is made up of amino acids as far as the amino acids are concerned you don't have to by heart the different types of amino acids that is present however one specific characteristic of gram negative jeneke olop agote kolo no gram positive bacteria ki thakibo teichoic acid gram negative bacteria ki thakibo depa so question ahe depa the presence of depa is a characteristic of which bacteria the presence of teichoic acid is a characteristic of which bacteria so out of all gram positive ot ki thake lysine yar por question nahe right hoday question to yar por ahe je depa kot ase depa is a characteristic of gram negative right whereas teichoic acid however it's not an amino acid right kino etu amino acid kino we are talking about the unique characteristics of gram positive and gram negative right next glycan is also made up of amino acid kintu alternating units ebar neg thakibo ebar nem thakibo akko ebar neg chain type right as far as neg and name is concerned detail or pura full form nalage neg aru name word gita mon rakhiba because we are going to talk about another type of bacteria hoy kintu neg ba name na thakibo pare so at that time we will be comparing je kisuman bacteria thake kisuman bacteria na thake so all the points that you remember is bacterial cell wall boli kole peptidoglycan right it is made up of peptidoglycan or unique point ki depa is a characteristic of gram negative right and neg and nem part of peptidoglycan that's all thank you so neg aru nem jete tomar eneke alternate ke thakibo neg nem ako neg etu backbone or nisna hobo noy spinal cord dalor nisna hobo noy so glycan forms the backbone of peptidoglycan is point understood if you want to write it down you can write it down forms the backbone of peptidoglycan clear for the next class staining in bacteria heading kori thoriba ba bacterial staining je likha thank you